I have to say I'm sad that I am not in person with all of you in Paris, but hopefully, we'll be able to get back to seeing each other before too long. And so today, what I wanted to do is to tell you a bit about some just preliminary data on a relatively new direction for my group, still looking at microbial communities, but from a bit of a different philosophical perspective. But just to get us all on the same page, I want to start by highlighting that the microbes are everywhere, every corner of the globe, in deep mine shafts and under the ice and these lakes. But not only that, they're also showing up in all sorts of cover of many of the publications that we read for other purposes. And the basic reason for this, of course, is just that it has become increasingly clear that microbes are playing a really essential role in human health. So although this has been the focus of much of the work that you see these days, I think it is important to remember that we've known for a long time that microbial activity is also playing an essential role in the health of the planet. Right? So for example, microbial metabolism is responsible for nearly half of carbon cycling in the ocean. Right? So we really want to understand these microbial communities and how they behave. And of course, the importance of microbes has led, well, at MIT, over the last couple of years, I have been serving as co-director for the microbiology graduate program, and 1 year for the program T-shirt, we had this image, where we said, microbes drive the world, and we're highlighting just the importance that microbes play for the health of the planet. But in reality, it's microbial communities, not individuals. So this image was not very accurate, because really it should look something more like this, right? Where you really have a collection of microbial species that are together, with their hands on the steering wheel, controlling the fate of the planet. And you can imagine that this leads to some consternation for the globe over here, basically because it's not clear how the function of these communities emerges from these tremendously complex set of interactions. Indeed, if you go and you look at, for example, human stool, what you see is that there are many, many different species of microbe, mostly bacteria, that of all shapes and sizes. These are false colors, but many different species. And not only are the species complex in the sense of many of them, but the interactions themselves are also complex. Right. So if we were to zoom in on this using some PowerPoint microscope, what you'd see is that the species are interacted to a lot of different ways. So of course, there's resource competition, but beyond that, there are many other kinds of interactions. There can be, for example, predation and the production of toxins that are killing other cells. Beyond these sorts of negative interactions, there are also going to be a variety of positive interactions. There could be some sort of cross-feeding, cross-protection. And in many cases, what we want to do is we want to compare the strength of those interspecies interactions that I'm telling you about with the strength of intraspecies interactions, the interactions within the species, because of course, even cells of the same species are also going to be competing with each other for food and other resources. All right, so this slide is starting to get full. It's complex, but things can be even in some ways worse, because in addition to all of the interactions I've told you about now, there can also be higher order interactions where the species are, the presence of one species is really fundamentally changing the nature of the interaction between two other species. Okay. All right, so given this complexity, it would be easy to maybe just throw up our hands and give up and acknowledge that we're never gonna understand anything. But I think that's overly fatalistic. So a lot of the approach that we take in my groups to try to understand, despite these potential complexities, can we nonetheless say something perhaps hopefully simple about the way that these communities are assembling and functioning. Right. And of course, we know that there are many cases where there are simple rules that govern otherwise what you might think of as complex systems. Early on in the scientific process, we discovered that gases actually follow some simple rules. We know that the pressure of a gas really is determined by just a few things, right? The number of number of molecules, the temperature, and so forth. And that works even when there are multiple gases together within your chamber. Indeed, it even works when it's not an ideal gas, right? So there are corrections associated with interactions and so forth between the molecules. Of course, that's a very weakly interacting system. And you might say, oh, well, of course, there are some simple things going on there. So it's nice to know that something like the flow of a fluid, that is an emergent kind of simple rule that governs actually quantitatively the way that a fluid flows. And that's true even though there's very strong interactions between the molecules within a fluid. And then finally, within the realm of chemistry, chemical biology, if you've ever taken organic chemistry, we often design chemical synthesis schemes by doing these sorts of arrow pushing. So like there's kind of these electrons and we draw arrows, say, okay, the electron kind of attacks this bond and then you can move things around. And this is a simple phenomenological scheme that we've developed that has no obvious relationship to the quantum mechanical description of electrons, but nonetheless defines a powerful layer of abstraction that you could use to actually design how you're going to make a molecule using chemical techniques in the test tube, right? So these are kind of different examples of how you can either get simple behaviors in these complex systems, or you can define a layer of abstraction to understand what's going on. And a lot of the work in my groups to try to figure out when is it that you can use similar kinds of approaches within the context of microbial communities, right? So just as a historical background, one of the early advances in this area was by Robert May in the early 60s, where he pointed out that if you have a complex community with many species, you can use ideas from random matrix theory to try to make predictions regarding when it is that that community will become unstable and will lead to extinction of some species. Right? So he was analyzing this classical model, the Lotka-Volterra competition model, where you basically just have where n is the population size, the change in the population size, and if we were to divide by n, this would be a per capita growth rate of the population. It's normalized to start at 1, and then it just decreases as a result of interactions between other species in the community. Or if the alphas are negative, there could be some positive interactions. But basically, the per capita growth rate is just altered in a linear fashion associated with the population sizes of the other species. And it turns out that if you just have random interactions between the species, then for large communities, the distribution of eigenvalues follows very simple form. And in particular, there's the kind of the distribution of eigenvalues will fall on a circle. And what you can do from that is you can say, all right, well, depending upon where those eigenvalues are going to fall, the community is either going to be stable or unstable. And basically, for the community to be stable, it requires all those eigenvalues to fall on, they all have to be negative. The real part has to be negative, right? The center of the circle starts at -1 on this kind of imaginary plane, right? So then what you can say is, based on the size of the circle, if the circle's big, then some of the eigenvalues end up on the positive side of the plane, right? And then you want to know how big is the circle? Is it bigger or smaller than 1? Because that's going to determine whether it falls to the right or not of this vertical of the central axis. Right, so the radius then is just determined by these two things. So there's the interaction strength, and there's the number of species. And all right, as shown here. Okay, all right. So the May bound, as they call it, is making a prediction that basically says, right, if communities are strongly interacting, you expect them to become unstable. You get some extinction, or if the community is complex, right, and some combination of those things. Right? I think these are really interesting ideas. Our goal is basically to explore this stuff experimentally and to ask, well, what happens beyond the May bound? All right. So in general, in my group, we have taken this bottom-up approach, but we first focused on single species, then we looked at pairs to build up synthetic communities. We also look at a variety of both natural communities outside of the lab and pseudo-natural communities within the lab. But over just the next 10 minutes, I wanted to tell you about our approach to trying to understand what goes on beyond the May bound. right? So basically, is it really the case that complex communities lose stability? And this is a little bit funny because we often talk about complex communities as being more stable, right? That's why we like diverse communities. So, we can talk about what's going on. Also, I'm going to address the question, well, what sets the strength of interactions between species within these microbial communities? And more generally, I guess I'm probing the question of which community dynamics don't depend on the details of the interactions between particular species. And when is it that we can just use a statistical description to try to make predictions regarding these communities? All right. So this is work that has been led by Jilong Hu, who's a graduate student in my group. And what he started out by doing is just to orient ourselves, he has been modeling this logical Volterra model, the same thing that I talked about before, but together with dispersal from a source community, right? So there's going to be some number S of species in a source community. There's going to be a low rate of dispersal of those species onto our focal community. And then we're going to ask, well, what happens within the focal community? Right. And consistent with like this May-bound, basically, if you have very small number of species or very weak interactions, right, down here at the bottom left, then you get stable coexistence of those species, right? So they survive and the dynamics are stable, right? Then as you move to the right or up, then you cross this region that you might call a boundary equilibrium, where it's still stable, but some of the species go extinct, right? So on this space of species, or you arrive at the edge where some of the species are basically going extinct, except for this little bit of of arrival from the source community, right? But still stable. And then as you go further to the up and to the right, then you enter a regime where you have persistent fluctuations that you generically would eventually expect to become chaotic. All right, so there's basically these three kind of regimes that emerge within this simple model. You can also plot on a color map the fraction of the original S species that are surviving, right? And again, what you have is that on the bottom left, all the species survive. Then through this boundary equilibrium, you get fewer and fewer of the species surviving. But interestingly, at the top right, about a third of the species survive. And that's roughly flat, where you get about the same fraction of the species surviving, independent of the number of species that start, as well as the strength of the species interactions. Okay. All right, so that's what you kind of get in a simple model. There's a lot that we are trying to understand together with our theoretical collaborators, so Martha Barbieri and Guy Boonen, who have been working on in this field, have really inspired us in terms of how we think about stuff, and they have been wonderful to talk to. And some of it's possible that some of you got a chance to hear Matthew speak, so you already appreciate some of the insight that he can give. Okay, so that's kind of where we're starting in terms of expectations from theories. And that question is, what can we do experimentally to make sense of this? All right, well, first of all, on this phase diagram, S is the number of species in that source community, and that's something that we can control experimentally, right? So what we've done is basically take collections of, we have hundreds of different isolates, mostly from the soil or from the gut of worms or other places found in nature. And so now we have a collection of species, and we can ourselves be the ones that are controlling this source community. And what we're going to do is just add dispersal to our focal communities and look to see what are the dynamics of the resulting communities. All right, so what we did is in this first experiment I'll show you, we basically took 24 different species, and then we assembled many, many different collections of 3, 6, 12, and then we did the all 24 species community. Right, and basically, if you go and you look at the biomass of these resulting communities, all right, this is just an example of one of the 3 species communities. What we found is that the 3 species communities were essentially always stable. And this is in the context of daily batch culture. Okay. Now, you can ask, well, what happens if you add 6 species? Okay, so this is a different, now this is now just one particular 6 species community. What you see is that here it's reaching a lower biomass. Not always the case that more species leads to lower biomass, but this one it did. Again, stable. So these are replicate communities. They're all stable, doing the same thing. Whereas what we found is that as we went up to 12 species, now what we're seeing are persistent fluctuations in the biomass of the community. And that's accompanied by persistent fluctuations in the relative abundance of the species as well. Right. So you can see that although these three communities all started out the same, we mixed them all together. Now, each one is independently fluctuating, and they're fluctuating in different ways. Okay. All right, and that was true for the 12 species as well as for the 24. All right, so this is a case where we can just control the number of species that are arriving in this focal community, and we can see that they start out being stable when they're simple, and then they're crossing some threshold, and they're losing stability, and they're becoming, they experience persistent fluctuations. Right, so we can combine all of our different combinations of the 3, 6, 12, and the 24 species community. What we see is that indeed the single species communities, they were all, I don't know, you can decide whether to call them communities, but the single species on their own were always stable, no fluctuations in biomass. The 3 species communities, almost all of them were stable. But then as we increase the number of species, what we observe is an increasing frequency of these persistent fluctuations within the community. All right, so this was in a particular environment, in a particular media, right? So in that sense, we're just moving kind of along horizontally along these phase diagrams. So then the question is, well, can we also move vertically in some way, right? And it turns out that actually, if you change the nutrient concentration, what we found is that you can change the strength of interactions between species. And that's basically because although in these models, we're imagining the species directly interacting with each other through some strength of inhibition and so forth, of course, the way that most microbial species are interacting is not directly, but really through some chemical pools in the environment, right? So each species is secreting different things and they're uptaking different things. And it's through that modification of the environment that is really mediating the interactions between the species. And one of the things that we've found over the last few years that is maybe not a surprise, but we found is just time and time again, one of the dominant modes of interaction is changes to the pH. So basically, if you have a lot of nutrients, then you have a lot of changes to the environment of a variety of sorts, including the pH, and that increases the strength of interspecies interactions. Okay, so the proposal then, or the possibility is that by increasing the concentration of nutrients in the media, we can move vertically on this diagram. Okay. And indeed, in a recent paper by Christophe Rotsky and Julien Barrère, two members of my group, we showed that by changing the nutrient concentrations, we could go from a situation where in blue, in low nutrients, there are weak interactions, we have stable communities. And then as we increase the nutrient concentration, we would get these persistent fluctuations. And we observed this in the context of natural communities propagated in the lab, right? So in this case, what Christophe did is he went and he got compost or flower pots or other kind of really kind of pseudo-natural environments with hundreds of species, and then he saw these things, right? So this is not controlling the number of species, but this is just taking lots of species to assemble truly complex communities. But he saw that indeed he could go from stable and actually stable high diversity communities to unstable low diversity communities by changing the nutrient concentration. So then the question is, can we utilize this in order to really map out that phase diagram? And it seems to work, right? So okay, again, so on this diagram, more species to the right, more nutrients is up above, and indeed, what we could do is assemble many communities. And in this case, we went up to 49 species. And what we showed is that basically in low nutrients, these were all stable. In intermediate nutrients, we started getting unstable, persistent fluctuations at around this 24 species mark, whereas at high nutrients, that's where it occurred around the 12 species mark. Okay, so what we can really see is that by changing, we can change both the number of species in that source community, as well as the nutrients change the interaction strength. And we can really start to see how you cross these different regimes at different points. All right, we are at 20 minutes, so I'll sum up. Right, so theory that has gone back for decades has predicted that there are, in principle, universal behaviors of these complex communities. And there's been a lot of work trying to understand, well, if you have correlations in your interaction matrix, what happens? Well, and it turns out that this circle of eigenvalues becomes an ellipse in one direction or another, depending on whether you have more predator-prey type interactions or mutualistic and so forth, okay? That would be an interesting direction to try to understand experimentally as well. What we've been finding is that these communities are losing stability either when we add species or when we add nutrients. So when we increase the complexity or we increase the interaction strength of between the species and the community. And finally, I think that this is just kind of a first step towards trying to understand possible, simple yet predictive kinds of understandings of the emergent properties of these complex communities. This is these still very early days on this project, so I'd love to get your feedback. We obviously, we're sequencing all these communities so that we can really probe how it is that they're fluctuating, and also see if we can measure that phase diagram of the fraction of the species that are surviving as a function of these parameters. And that is that's ongoing. We're waiting for those results to come back. So yeah, with that, I will thank the group. So this, as I said, was work that was primarily led by Jiliang Hu, together with Danny Moore, who I seem to have, oh, there, okay, so Danny is here, and I'm happy to take any questions.
Thank you, Jeff. That was really cool. We have a question, let me have several. So one question is from Denny. Does this imply that the less nutrient